about um, these structures. Collagen bundles, beautiful. So if these are collagen bundles, that makes these cells what?
Fibroblasts.
Fibroblasts. Look at the euchromatin. Very little heterochromatin and lots of euchromatin. Besides, they have the orthodox appearance, like enlarged. Name the tissue in the main field. Jeffrey, what did you say?
No, I just repeated what you said.
The tissue in the main field. Come on, you can do this, man. Make me proud.
I disappoint you.
The tissue, just the tissue. I didn't say be specific or anything. Remember that this is very easy because at this point, you just have to narrow it to two different tissues. Is it muscle or is it connective tissue? And then, well, this is not muscle for sure, right? So it is connective tissue. So, okay, which type of connective tissue we have? Is it fat? No, right? Is it dense regular?
No.
No. Is it loose areolar?
No.
Cartilage?
No.
So what else could it be?
Mucus.
Do you see mucus over there?
Wait,
In the main field? No.
No?
These are bone spicules, my friend. This is bone. Bones. These are the trabeculas of bones. This is kind of like a low power magnification of a bone of the skull.
Wait, so these are the plates?
And that bone is forming from scratch. So you don't see any cartilage in there. You see the mesenchymal tissue is turning into bone. Like you don't see any endochondral figure over there because bones of the skull and bones of the vertebrae, they form from scratch into bone. Those are the first bones that are ever formed in the body. They have to form first from scratch because the brain needs to be protected. So the brain is forming so fast, it cannot wait until a bone goes from cartilage to bone. No. It needs to go as fast as the brain and spinal cord are flat. So that's why the skull and the spinal cord, the vertebrae, they're all formed by intramembranous ossification. Everything makes sense. So these structures are called bony spicules. And this is a figure of intramembranous ossification. Bone is forming from scratch from mesenchymal tissue. Lots of blood supply in the area. So now different to this, you see this? Now you get to see a figure of endochondral ossification because you see cartilage turning into bone.
Yeah.
But over here you didn't see cartilage at all. Name this cell over here. This is something that you either open garnet and hire or you didn't. Because you will not guess what the cell is unless you see it. You have seen it and it's coming from your lab book. So have you seen that cell? Which page is that one, Miguel?
93.
Page 92 on your garden center, you need to see all those pictures. Because that's your lab manual, right? This is the third game. I'm using your lab books. So that is a chondrocyte. They look like heights. I remember when I was a student, I used that same book. 15 years, more than 17 years ago. That same book, of course, it was a prior edition. And when I studied those, they looked to me like kites. And I never forgot. And I said, when I have my own students, I'm going to teach them the kites. So there's a lot of information here. This is an electron micrograph. And remember the territorial matrix that I told you? Look at the fibers of the polymerized glycosaminoglycans right here. No wonder why it stays so dense, because these fibers are really dense to be seen at the molecular level in an electron micrograph. Look at them around. And of course, the cell has all kinds of organelles, just like any other cell. So this, technically speaking, it's inside the lacuna. So that's territorial matrix. Or chondroblast. Actually, it is a chondroblast. It is actually a chondroblast. It is a blast. I don't know how they say it over there, but the truth of the matter is that it's inside a lacuna and it divides. You see, they say chondrocytes, but they are technically chondroblasts. Look at the euchromatin versus hetero, and the extensive amount of raw PR. So they are dividing. So, books have their own glitches. So if you put chondrocyte/chondroblast, perfectly fine. You cover the two bases. But since I want to teach you, they are chondroblast. And the proof is in the pudding. Right there. Look at that euchromatin. So how about this one over here? The A cell.
Oh. It's eating the bone. So
So
chondroblast.
it is.
Uh,
If it's eating the bone, then it's
exact class.
an adipocyte, right?
Osteoclast.
Osteoclast. Very good. Osteoclast. I don't know if you get to see the multiple nuclei.
Little bit.
Then the B is
Dehydration zone?
the what?
Hydration zone? Or the ruffle border?
Ruffle border, hydration shell,
Shell.
clearing zone. They all refer to the same thing. Now, the one thing you're forbidden to say is osteoid.
Osteo what?
Osteoid. That you're forbidden to say that because you see, this is osteoid over here. Because that is an osteoclast. But you either eat the bone or you form the bone.
Okay.
So I don't know if you pay attention, this bone is being formed, and that's why there's deposition of the collagen over here, and that's why this part over here looks darker. As opposed to here, the clearance zone is reabsorbing and looks lighter. That's why they call it clear in, because it's clear. There's a lot of acids over there. Okay? Acids coming from lysosomes. They're released outside, and that acid eats the bone and metabolizes. So, name structures indicated by the letter H.